We're going to be talking about one of my people I most admire, who's Gregor Mendel. And turns out somebody figured out he was a really smart kid. And so they thought, but he was really, really poor. So they're like, we got to get this kid an education. But he's really poor. He lived in what was then the Austrian Empire. Now it's part of the Czech Republic. And so they said, well, the only way we can get this kid a free education is if he becomes a brother. So he became an Augustinian monk. So that's how he ended up being a monk. And he, after his experiments, which were very successful, and the things that apparently he liked doing most in his life, they made him an administrator, so he quit doing experiments and lived out his days not being very happy. But so I'll tell you the happy parts of his life as soon as we start here in about 15 seconds. One of the things that happened that we're going to try to avoid in this century is that people almost completely forgot about his work. I don't know if you've heard that story. So for about 40 years, this paper came out. It was hardly read. Almost everybody forgot about it. And so what we're going to try to do in this century is we're generating a lot of data, but we're going to try to make it so people don't forget about it and it's accessible. And that's what a lot of this whole unit is going to be about, is keeping the data accessible and using bioinformatic tools. So that's what I'm going to try to do. And before I get into the real deal parts of the lecture, let me talk about stuff that you would need to be reading here fairly shortly for this all to make sense. First of all, in this second part of the reader, I've given you 2 chapters from a book, chapters 5 and 6, and they do just a great job of talking about things that I'm going to be talking about in this lecture. Such things as qualitative versus quantitative traits and patterns of heritability and such as that. They do a great job. Also, there is this paper by Judith Grisel in your reader, and here she's going to talk about quantitative trait locus analysis, which is the centerpiece of this whole part. And she does a great job in just a very few pages of describing one of the things we're going to be doing, in fact, the centerpiece of this whole unit. So read those papers, and also in there, there's a paper by Williams et al., and that's the experiment that we're going to try to replicate in lab, okay? So you won't need to be conversant with that quite yet, but first, please get through those 2 chapters in this article by Grisel, and hopefully things will make more sense in lab. By the way, and they're super easy reading. I mean, they just do a great job of making it very accessible and wonderful articles. Okay, so things I want you to get out of this unit. What we're going to do, as I mentioned earlier, is we're going to talk about bioinformatic tools, ways of hanging on to data and making it accessible. We're going to introduce QTL analysis. QTL analysis stands for quantitative trait locus analysis, and it is literally almost true. Almost every talk I go to now has a little QTL analysis in it. What it is ways for figuring out how variations in phenotype can be linked to loci on chromosomes where the genes that influence those phenotypes are located. Okay? It's a fairly new technique, probably about a decade old now, and as I initially started to get this ready in the morning, I went to a talk on I think it was autism. So I did QTL analysis on autism, figuring out which genes it was, and then later in the day I went to another talk on gay versus straight. Is there a genetic basis? And they were doing a QTL analysis. Last week I went to another talk on fat genes. They're doing a QTL analysis trying to figure out where fat genes are on the chromosome and what they might be. So it's a happening technique. People are using it a lot. You're going to hear about it a lot in your lifetime. As I stand here today, last I heard there were 4 genes that had been positively identified with QTL, but people who are detractors of this method say, oh, it's worthless, only 4 genes, blah, blah, blah. But it's kind of like the people who are standing there with the Wright brothers saying, oh, it only went a few hundred feet, the thing's worthless. So it's a technique that's is getting better even as I speak, and you're going to be hearing about it. To talk about that, we're going to have a tiny review of genetics, which you could probably do even better than I can. We're going to talk about and review some molecular techniques, some of which Dr. Bolter taught you about, and some others. Have you guys heard about in situ hybridization? Okay, so you have heard about that. Gene chips. Okay, so you've heard about at least some of these. Great. And we're going to get into statistical analyses and concepts. The good news for you is we use computers, so you don't have to do all the hairy, nasty calculations that I had to. But the conceptual stuff is not that bad. All right, so that's what we're going to do in this unit. And so here is how we're going to do it. Back in lab, what we're going to do is, first of all, quantify a phenotype. And the phenotype that we've selected is the olfactory bulb. Not because it's perhaps the most interesting part of the whole brain, but because I can get all of you to pretty much agree what's olfactory bulb and what's not. And one of the things we need to do is have people do things consistency to keep the error variance down. And that's going to be one of the goals in this unit, is to get rid of error variance. Okay, so one of the problems that we'll be solving tomorrow is the brains got shrunk when they got cut. And so we're going to have to reverse that mathematically. They got differentially shrunk. What we're also going to do is there's going to be two raters. So there's going to be, you're going to have a set of brains to take care of, and somebody else in lab, unbeknownst to you, is going to have the same set. And so it's kind of a check on each other. If you guys don't agree, next Thursday, by the way, next Thursday is required lecture down in the lab, and we're going to sit you all down. If you don't agree with your mystery partner, you guys got to sit down, hammer it out, and figure out why you're discrepant and get that solved, okay? So that's the multiple rater things. We've got extraneous variables. So these mice come in all kinds of sexes, age, body weights, and other kinds of flavors. And that, of course, increases the variance in your phenotype. We got to take care of that, all right? And we will do that with statistics. All right, and then once we get all the data all cleaned up, what we're going to do is we're going to take the variance in the data and then link it to chromosomal markers. We're going to, in other words, do a QTL analysis. And the way, so this is one of the endpoints you're going to be looking at. So I'm going to show you the endpoints now, and then we'll get back to them, and hopefully they'll make sense. So we're going to be doing this called a likelihood ratio statistic as a function of all these different markers. And what we have here is a graph of all the different chromosomes in a mouse laid end to end to end to end to end, 1 through 19 and X. Okay? And so when we get peaks here, that suggests that, say here we got a peak on chromosome 6, which is something we often do, that on that region of chromosome 6, there are genes that influence our phenotype. Okay? So that's what we're going to be heading for, and I'll give you more details on this QTL analysis as we go through this lecture. All right? Once we find these peaks, we're going to start looking for genes that are at those peaks. So that's a hunk of chromosome. There's a whole pile of genes under that hunk, or that compose that hunk, and we want to know what are those genes. And the way we're going to do that is we're going to use the UC Santa Cruz genome browser and go in there, and then you get these lists of known genes that are on that stretch of chromosome. And you can go through, and what we're going to do is we're going to look at those genes, have you sort through those genes, and find a gene that is actually highly expressed. Forget this down-regulated thing. We're going to look for one that's highly expressed, and that data already exists. So people have already run the gene chips and figured out for that gene, we want to find one that's highly expressed. And you'll also find that on the UC Santa Cruz genome browser. And this isn't projecting worth a darn. This particular gene that I looked up, ASXL2, turned out to be, there should be a red box right there in the striatum, which means it's highly expressed. It unfortunately was a bust for olfactory bulb, and that's what we're going to be interested in, ones that are highly expressed in olfactory bulbs. Okay, and then what we're going to do is have you look at that gene that you find that is highly expressed in olfactory bulbs in depth. And so one of the things that we're going to ask is to use yet another bioinformatics tool. This is the Allen Brain Atlas. You all seen this thing at all? All right. So what the Allen Brain Atlas is an app. So you'll get some work with an atlas, a map of a mouse brain, and then it will also show you by means of in situ hybridization what cell layers in the olfactory bulb express your gene. Okay. So we'll figure out what cell layers the gene is expressed in using that tool. We're going to go to back to NCBI like you did in Dr. Bolter's module, get and find out what the sequence is and the coding sequence is. And as you also have done in Dr. Bolter's module, you go to PubMed, find an article about that gene, and write us a little summary of it. Of course, you all have gone to PubMed before, I trust. Okay, excellent. And so that's what we're up to. So that's the sort of things we're going to be doing. So what I'm going to do in this lecture is define some terms, describe the phenotype a little bit at the end, and talk about recombinant inbred strains from which our phenotype will be measured. So recombinant inbred strains are key to this, and talk about chromosome mapping. So this I hope you all took genetics, right? That's LS3, 4? 3 or 4? 4, thanks. All right, so phenotype, okay, is any observable characteristic or trait of an organism, the morphology, development, biochemistry, the physiology, properties, or behavior. Okay, so that's something that people often forget. You know, you think about phenotypes as hair color or nose size or something like that, or height, but behavior can be a phenotype too. The genotype is the exact genetic makeup. Okay, if two organisms differ even at one locus, they have different genotypes. Okay, but this ought to be stuff that's very familiar to you. So we're going to talk a little bit. Gregor here is going to get us through some genetics. And what I want to start out with is talking about phenotypes and contrasting qualitative traits with quantitative traits. And back when you took LS4, probably a lot of what you talked about were qualitative traits, red versus white eyes, stubby wings versus big wings, things like that, because they're really nice and easy to study. They tend to be influenced by a single gene. They follow simple patterns of inheritance. Phenotypes fall into distinct categories. You can say, okay, they got red eyes or white eyes, and the trait is typically unaffected by the environment. And truly, almost no trait that you can think of with regard to humans is a qualitative trait. Okay, almost all of our traits that you can think of are quantitative traits, things that are influenced by multiple genes, perhaps interacting genes, don't follow simple patterns of inheritance. The phenotype tends to be measured on a continuous scale, things like height or IQ or things like that, and the trait can be expressed by the environment. So most traits that we think of are actually quantitative traits. They're harder to get a handle on. And so one of them that I've already mentioned is something like IQ. So you could have something even like a mental trait, which could be a phenotype. And this one, actually, people have come to think is influenced a good deal. When I was your age, my professor said, it's all environment. And then I got into this literature and found out they weren't telling me the real story about even the people who think environment's important, they think about half of it is due to genes, as you can see here in this table. So biological and psychological traits tend to be normally distributed, polymorphic, influenced by several alleles, possibly several genes. Okay, so they're, again, quantitative traits. There is, I only could think of one instance where there's a qualitative trait in humans, and that's Huntington's disease, with which you are familiar or no? Okay, so what's the big symptom in Huntington's disease? Okay, so, and it actually used to be called Huntington's chorea, right? And so they show, yeah, honest. And before that, way back in Europe, it was called St. Vitus's dance. Why? Because those that are afflicted show these kind of dance-like uncontrollable movements, okay? It's a disease of the basal ganglia. And it does show a Mendelian pattern of inheritance. If you get the diseased allele, you only need to get one copy, and it will be completely penetrant. Okay, so you get one copy, it's dominant, you get the disease. Fortunately, it's very rare, okay, and we actually even know it's on the tippy end of the 4th chromosome. We don't know what the protein does in health. We know what happens when it's screwed up. And if it's got too many, if the gene has got too many trinucleotide repeats, that's the pathological allele. But other than that, there's not a whole lot of characteristics that aren't quantitative traits in humans. Okay, let's talk about alleles. All right, so it's a member of a pair of a series of different forms of a gene. In a population, you can have lots of different alleles, but in an individual, how many different forms of an allele can you have? 2. 1 on each chromosome, right? Okay, so that's the max. You could be homozygous, right? Okay. Hang on to those ideas. Okay, so what we're going to do is we are going to recombine DNA. And I would not recommend this as a pickup line, but the way that we're going to recombine DNA is we're not going to do it in a tube like Dr. Bolter, but we're going to ask these mice to fall in love and do it for us, okay? And so I'm going to get back to that story, but let's first talk about recombining DNA. So good old Gregor here says he never saw any evidence of recombination, okay? And so he came up with some laws, one of which was the segregation of alleles and independent assortment of genes. This ought to be familiar to you? Yeah? Good. All right. And so he did experiments such as this, where he took an F0 generation that had a highly discrepant trait, here wrinkled versus smooth peas, and in the F1 generation, everyone showed the same phenotype, even though everyone was heterozygous, right? And then what he did was he crossed the F1s and got this split there of 3 smooth to 1 dented, and so that's sort of the Mendelian pattern of inheritance. So he says, okay, what evidence is there for recombination? So let's think about what if we had 2 genes, and Mendel was right. Okay, so we have a gene P, which is pubert or hairy, and D, which is for dwarfs. Okay. We got 2 genes. If Mendel's right, how are these plants going to come out? Actually, it's going to be 1/4, 1/4, 1/4, 1/4 if all those genes are completely independent, right? All right, so that's what Mendel would predict. Now, what if those genes were on the same chromosome? Could you have an independent assortment of genes? No. All right. So if they're on the same chromosomes, we now know you would predict that you would get half of the plants being smooth and tall and half the plants being hairy dwarfs, right? You'd have a half-half split. All right. Now, drum roll, please. How does it come out? Let me tell you. These genes are going to be on the same chromosome. How does it really come out? Does it come out half-half? Doesn't? Come out 1/4, 1/4, 1/4? Doesn't do that either? Well, you got your money's worth when you took genetics. All right. It doesn't quite come out half-half. Gets close. Gets close. 48% are smooth and tall, and 48% are hairy dwarfs. But you get these 2% that are combinations. Why is that? Recombination. Exactly. So, what happened there? In a few instances, when the chromosomes get together to make the gametes, they cross over and recombine the DNA. All right? This is a story that you all know already? Okay. Excellent. And so we're actually going to ask our mice to do the same sort of thing for us and mix up their DNA for us by means of this recombination. So during meiosis, the homologous chromosomes pair up in these things called chiasmata. Looks like X's, so that's why it's a chiasmata. And the chromosome from the mom and the dad get back together. When you make the gametes, they can cross over and swap and recombine the DNA. All right? This is a story you already know? Yes or no? Yep, okay, excellent. You guys are way ahead of this game. All right. So now the gametes of the F1 generation have some of the DNA from each of the F0 strain. Okay, does that make sense? All right. And so they're going to end up with a collage. All right. So now let's talk about how to make a recombinant inbred strain of mouse. So we're going to start with two already inbred mouse strains. So the inbred mouse strains are homozygous, right? They've been inbred for generations upon generations. Say yes. Yes. Okay. And so they're homozygous, and we're going to pick a couple of strains that are really discrepant, far apart on the phenotype of interest. Okay. And we're going to cross them. So the strains that we're going to use are actually these two. So this is going to be the daddy strain. This is the DBA mouse, which we will call the D mouse from now on. And the mommy F0s are the C57BL/6Js, okay? Little black mouse. So we'll call this the B mouse, okay, and this the D mouse from now on, all right? So they're the F0s, and we know that they are really discrepant on our phenotype of interest. And our phenotype of interest is going to be olfactory bulbs. So our C57BL/6J, the B strain, are less sensitive to odors. They can't seem to smell things as well as the DBAs, and they have smaller olfactory bulbs. We already know this, so they're discrepant on the phenotype of interest. That's key. All right? Okay. So now we're going to put them together and make an F1 generation, and then the F2 generation will have chromosomes with DNA that are combinations of the F0s. Okay? It's going to be much like what Gregor did with the P's. All right, so let's look. I'm going to go to this slide. This slide is the key to most of this module. You understand this slide, you're going to understand how we got to our mice. All right, so we're going to start here. This is the F0 female, the B mouse, and this is the male, the D mouse. And we're going to look at one chromosome just by way of example, all right? And this one chromosome here, we're going to represent hers as being solid blue and his as being solid red. And it's solid blue because are they homozygous? Yes. So all of the alleles for this particular one are the same, both chromosomes. Check? Check. Ditto for this one. All the alleles are the same. So we cross these two, we get an F1 generation, okay? And the F1 generation, everyone has the same genotype, right? Got one chromosome from mom, one chromosome from dad. Now, when these guys go to make their gametes, then the chromosome from mom and the chromosome from dad are going to get together, and in some instances, there'll be some crossing over and some recombination, right? Is that, by the way, a common or rare occurrence? It's relatively rare, okay. And does it happen with big hunks of DNA or tiny little hunks of DNA all shuffled together? Tends to be pretty big hunks, right? Not, you don't get kind of like a random shuffle like with the cards or something like that. Pretty big hunks of DNA all cross over and swap. Okay, so then we end up, the first F2 generation, as you can see, has all different kinds of recombinations of the F0 DNA. So that's what this represents here. These two chromosomes are now all kinds of different kinds of the recombinations of the F0s. All right? But as you can see, like this particular animal, let's say there's a gene right on the tippy end here. Are they going to be homozygous or heterozygous? They're going to be heterozygous. We don't want that. All right? So we want everyone to be homozygous for a given allele. All right? How are we going to get them to be homozygous? We can't make them be homozygous, but how about their offspring? Can we make them homozygous? How? Inbreed them. Okay, so you take brother-sister pairs and start tossing them together, and they're mice. Okay, so when it's time, it's time. And so you do that for 20 generations, and what you get then is for when it locks into a given allele, you can't get back to the heterozygous state, right? So you do that for 20 generations, and for a given allele, they'll be homozygous. All right? But they will still be a combination of the F0 DNA. All right? It's just that every allele, some of their alleles will be from this strain, some will be from that strain. Every gene will be homozygous, but across their chromosome, they're going to have this kind of collage. Some came from the D strain, some came from the B strain. Yeah? Is it impossible? Yeah. Like some won't? Well, how come there's crossing over in it's when these guys go to make their gametes, so that's when you get the crossing over. Let me ask you this question. Do you get a crossing over in this generation? Yeah. Exactly. Okay. So yeah, you always get the crossing over. All right. But when these guys do it, it's going to recombine the DNA for us. All right. And yeah, you'll get it back in these generations, but finally, when you get them to be homozygous for a given allele, they'll lock in there. All right. So you'll get stretches that belong to the original F0 mom strain and stretches that belong to the original F0 dad strain. Okay? All right. And what we're going to do when we do this is we're going to get all kinds of different combinations of that F0 DNA. So some of these recombinant inbred strains would look like this. They'll have the mom strain, the dad strain, the mom strain, the dad strain. And so this one will have a different pattern. And then another one will have a different pattern. And another one will have a different pattern. Okay? We can keep that within a strain, have the same pattern, but across strains, they'll have really different patterns. Does that make sense? I'm seeing not a lot of heads nodding. It's okay? All right. So what we're going to end up with is about 33 different flavors of recombination. And they're going to be recombinant inbred strains. Okay? And they will each have a unique recombination of the F0 DNA. All right? And so that's how we're going to attack this problem. All right. So I've already said this, but the inbreeding reduces the heterozygosity. If we inbreed them, the alleles for a given gene will become homozygous, and once they are homozygous, they're trapped and they can't go back to the heterozygous state. Okay, so what we could then do is we could figure out at given loci if that given recombinant inbred strain has the B form or the D form of the DNA. Okay? And so there are certain things that are markers that are known to be different between the B form and the D form. Okay? So, and this is actually one of these. This is not projecting real well, but this is something that ought to look darn familiar to you by now. All right, here's the molecular weight ladder, and this is the F0B mouse, and this is the F0D. So this is a little microsatellite, which tend to have variable nucleotide repeat sequences, and they're different with regard to this in this little microsatellite of DNA. All right, and so the D form is a little bit lighter than the B form. All right, and what you're seeing here on all these wells are they loaded up all these recombinant inbred strains, but this is the first F2 generation, okay, that we're seeing up here on this particular gel, the first F2s, all right. Some of them you can see have already, for this particular locus, have already locked into the B or the D form, but you can see some have both, All right? So they have both alleles. All right? So this is the very first F2 generation. After 20 generations, what would this gel look like? They're all B or D. They'll lock in one way or the other. Across all these different strains, would they all be B or would they all be D? No. Some are going to be E, some are going to be D. Right? Okay. All right. This is just a table that shows you as you go through generations of inbreeding, you knock out your heterozygosity, but you guys already know that. All right. We get rid of the heterozygosity. We breed brother-sister pairs together for 20 generations so that at any given locus, they either have the B form or the D form. All right, and we're going to have, as I mentioned, about 33 of these different recombinant inbred strains, and we'll have several individuals that will represent each strain. Okay? All right, so now Gregor says he needs a map to figure all of this out. So let's talk a little bit about mapping chromosomes. All right, there are 3 maps right now, and one is just mapping whole chromosomes by means of banding. Did you guys talk about banding, or is that like completely gone now? Never heard of it. Oh, because it's still used. It's still used clinically. Okay, so there's this GIMSA banding, which bands AT-rich regions. They used to use a stain, but now you can use it with fluorescent markers. And what you can use that for, this is a little sidebar that's got nothing to do with our study, but it is still used. And so what if you got a baby that might have Down syndrome? All right, baby's born, looks like it might be a Down's baby. How would you figure it out? Look for chromosome 21. So what you do is this kind of a stain, which they can do real quick and easy and cheap in a lab, and then look at the chromosomes and see if the kid's got 3 21s. Okay, then it's Down syndrome for sure. So it's still used. You know, these things were invented a long time ago, but they're still used clinically. What we are going to be using in our study is what's called a linkage map and a physical map. So what I want to talk a little bit now about is the linkage map. And as we mentioned, we've talked already about crossing over and swapping DNA and how it was first discovered there with peas and such as that. And when people discovered this phenomenon, there was somebody who had this incredible insight. And this guy's name was Alfred Henry Sturvetant, and his mentor was Thomas Hunt Morgan, who you may have heard about when you looked at the fruit flies and all that. And this Alfred Henry let's see if I can get his name, Sturtevant, figured out that if we can figure out the frequency of seeing these things becoming unlinked or linked together, we could actually get a map of the chromosome. Right? It was an incredible insight. He was an undergraduate when he figured this out. And his mentor got a Nobel Prize. And he actually went on to have a great career in genetics too. But he was an undergraduate when he figured it out. So what they did was they started making these great tables, figuring out, oh, how often do these traits become unlinked or stay linked together? If say you got 2 alleles that code for a given trait, and they almost always go together. They're going to be close on the chromosome or highly discrepant in space. They're going to be close, okay? If they can get split up fairly frequently, you would expect them to be far apart from each other, all right? So they actually made maps like this. And this map that we use, a linkage map, is in centimorgans in honor of Morgan, actually, because this undergraduate named it for his mentor. So a centimorgan is a 1% probability of genes becoming unlinked. Okay, so one way in which we express distances on a chromosome is in terms of centimorgans. And this paper of this experiment that we're going to try to replicate, this paper was published shortly before the mouse genome was completely sequenced. So their data is in centimorgans. All right. Now almost everybody, since the mouse genome has been completely sequenced, we usually talk about things being in megabases. All right. But there are conversions, and you're going to get a conversion table. You can go from megabases to centimorgans or centimorgans back to megabases. Okay, that's all been worked out. So we're actually going to have to figure out our data ultimately in centimorgans to see if it's comparable to theirs. All right, even though mostly people don't do it. Actually, Rick, do people do that? Is it all in centimorgans now? You've published in this field. No, centimorgans are a thing of the past, alas. Okay, but at any rate, for our purposes, we got to see if our data compares to their data. We got to get our data back to centimorgans, which we shall do. It will be easy. All right. Okay. So another way that these chromosomes have been mapped is to go along, as I mentioned, and figure out, okay, if there are places where the B and D DNA differ. Okay, and so they have found places where they do differ. All right, and so in fact, last I knew there were about 700, or there are more now. 4,000. We're not going to get all 4,000 in our analysis, I don't think. It's 4,000. Okay, so across the whole genome, all right, there are now 4,000 known markers where these two strains differ. All right, reliably. Known? Oh, right, but these two strains is our game, so we're just going to get them, okay? Yeah. So that's another way to map these chromosomes, and that is actually what this QTL analysis is going to use. And as I mentioned at the start of this lecture, what we're going to end up with is analysis that will allow us to figure out this statistic. If this statistic is high, we're at or near some genes that influence the phenotype. And what we're going to do is look at this statistic as a function of all these different markers across all these different chromosomes, 1 to 19 and X. And this is as if chromosome 1 was laid end to end with 2, 3, 4, up to 19 and X. Okay? Now, ladies and gentlemen, you notice there's no Y up there, right? Why not? All right, this is all dependent on recombination, right? Okay, how about the good old Y chromosome? Does it recombine? No, there's actually a little tiny hunk of it called the pseudoautosomal region that will, but most of it won't. Okay, X's, do they recombine? Yes. All right, so no Y in this analysis. All right, so anything on the Y chromosome, too bad. We don't get to find out about it. All right, well, I have this slide up. Let me talk about a little bit about what this analysis is going to do. So, do I have another slide coming up here shortly? Yeah. Let's jump to that. Okay, so as you can see, at a given locus, some of these recombinant inbred strains will have one form. These of a given allele, these guys would have the D form here for this marker. These guys will have the B form. These guys have the D form. So some will have one form at a given locus. Some will have another. Check? Does that make sense? All right. So what this analysis essentially does is it goes to a marker, all right, that we know is discrepant between B's and D's and says, and by the way, the other good news is some poor molecular biologist with a sore thumb has figured out across all these chromosomes for all these recombinant inbred strains which ones have the B marker and which ones have the D marker at all these different loci. Okay? Already been done for us. That's so cool because that was a pile of work. So it goes to the first marker and says, okay, all you mice, what we're going to do is say, okay, all you mice with the B marker, your olfactory bulbs are over in this pile. All you with the D marker, your olfactory bulbs are in this pile. Then we're going to look at the olfactory bulbs and see, is the phenotype different? Okay? And in this case, the phenotype didn't come out very different. So then we say, oh, there can't be any genes around there that have a big impact on the phenotype. Does that make sense? Then it marches on to the next marker. Does the same thing. All right, all you with the B form over here, all you with the D form over here, would compare the two piles and see if they're similar or different. It wasn't very different, on and on and on, through all 4,000 of these markers, okay, which this does in about somewhat under 30 seconds. And in this particular run, and often we get this result, we get a peak on chromosome 6 in this case, all right? What that, by the way, I should point out, if this blue squiggly line is the likelihood ratio statistic, if it gets over this green horizontal line, that is suggested. So it's suggested that we have likelihood ratio statistics that indicate there may be some genes there that are important. If it gets over that blue line, that's significant, and that really strongly suggests that there are some genes that influence the phenotype. Usually, we don't get it to be significant for reasons I'll explain in a later lecture, but we can often pop it up over that suggested line. All right, and often we find a peak over chromosome 6. I'm not saying you will. I expect that you will, but you never can tell. So what this particular graph would say is, okay, there on chromosome 6, there ought to be some genes that influence the phenotype. All right, so that's where we're going to start one of our stories in the second week of Latin. Okay. All right. So I think I am just going to point out we have, once again, we're going to have a whole bunch of recombinant inbred strains. We have, I believe, 33. Each recombinant inbred strain has a unique combination of the F0 DNA. Within a recombinant inbred strain, all the alleles and all the markers are going to be homozygous. So everybody's clear about that? Okay. All right. And we've already done QTL. All right, so now the phenotype. So Gregor's got to take a break here, and we shall shortly. So what we're going to do is we're going to measure the phenotype. All right, and our phenotype is going to be olfactory bulbs. Here's the other nice thing about this particular study. We are going to have a whole pile of mice to measure, but you guys don't have to cut and stain the brains because it's already been done by some other people who have sore thumbs at the University of Kentucky. Okay, so they already cut all these mice, and so everyone here took neuroanatomy, right? Any? Oh, excellent. Okay. Should I quiz you? Which way is rostral on this slide? Up. Very good. Okay, so, and what plane were these cut in? Should we make this multiple choice? Sagittal, coronal, horizontal. Horizontal. Okay, so rostral is up this way. These brains were all cut like this, okay, and stained with thionin, so they were stained for a Nissl stain. What's Nissl? You got to yell at me, I'm half deaf. It's a cell stain, and what's when in the cell, what's it stain? Yes. And in those cell bodies, what does it stain? Not many people know this. What is Nissl? No. No. It's the endoplasmic reticulum. Okay. Sorry, that was a trick question. That's not actually widely known. Okay, so this was stained with Nissl, so where it's dark, really dark here back in the cerebellum, you have very dense cell bodies. What's this job here? Say it. No. Sorry, I can't read lips. Okay, that's the hippocampus. All right. But at any rate, here's the olfactory bulb. So olfactory bulbs on these guys, I don't know if you guys have ever worked with mice, but they have to hang around and smell everything. So this is as if the brain were cut in sagittal, okay? All of this thing up here is the olfactory bulb, and here's the rest of the telencephalon. So we're going through, the first section is like through like this. The second section is starting to catch the olfactory bulb there, and then you can see there's finally a place where you're going to go through and there's no olfactory bulb left. Okay? So what you're going to have to do in lab is we're going to have to quantify the area of these olfactory bulbs, and we also know how far apart these sections are. So we'll be able to get to a volume. All right? So we're going to work on that in lab. So you get several mice in lab, Tuesday and Wednesday. Here's a close-up picture. There are the olfactory bulbs once again. And we'll use a NIH image. Anyone ever use this one? Okay, it's easy. If you ever use the drawing program, you can use this. And we'll quantify the size of the olfactory bulb. Okay, so what did we do in this lecture? We defined some terms. I described recombinant inbred strains to you from which our phenotype is going to be measured. You're going to measure the phenotype. We talked a little bit about chromosome mapping, and I told you a little bit about the phenotype. And for those of you who came to lecture today, do you have the papers? All right, so you can get your first assignment back. Don't forget, your second assignment is due Thursday, and lab is down in A456, and next lecture down in A456. And I'll be seeing you there shortly.